Greetings everyone, I am Ishita of class 8th from Queens Valley School. Today I am going to explain the laws and the mechanism of PCR machines. The polymerized chain reaction machine or thermocycler is a highly efficient tool used to amplify small segments of DNA or RNA. PCR systems are sought after systems for many studies, including testing for viral infections such as COVID-19. What does a PCR machine or thermocycler do? A polymerized chain reaction machine is commonly used to make millions of copies of initial small segment of DNA. It is also referred to as molecular photocopying and allows scientists to amplify DNA enough to study in detail. This is because analysis of DNA requires significant amounts of any given sample. A single piece of DNA is nearly impossible to work with for molecular and genetic analysis, so PCR amplification is essential. The amplified DNA produced by PCR has a variety of uses. These uses can include the investigation of genetic diseases, DNA fingerprinting, and the detection of bacteria or viruses. PCR is an integral part of much biomedical and research criminal forensics. How PCR works. PCR setup. The initial DNA sample which is to be copied, primers which are small sections of DNA which initiate the PCR reaction and bind to the DNA you intend to copy, a heat-resistant polymerase enzyme, DNA nucleotide bases, a buffering agent and salts required to create the ideal conditions for the reaction. PCR process. The polymerase chain reaction process can be broken down into 3 stages. Denaturing. The DNA template is heated to separate it into 2 pieces of single-strand DNA which happens between 94 to 95 degrees Celsius. Annealing. The temperature is lowered, enabling the primers to bind to DNA and initiate polymerization, which happens between 50 to 56 degrees Celsius. Extending. The temperature is raised again, and using the original strands as templates, a new strand of DNA is made by an enzyme, which happens at 72 degrees Celsius. Types of PCR machines. In addition to the standard methods of PCR, several modifications have been developed. Real-time PCR is a modified version of standard PCR. Real-time RT-PCR is used to detect viruses like COVID-19, causing SARS-CoV-2 very accurately, and now a day every viral and bacterial diagnosis is done by real-time PCR. Thank you.